Hi, my name is Caroline Jozefcyk, and I'm an antimicrobial stewardship pharmacist at Ohio Health, Mansfield, and Shelby hospitals. This presentation will cover the pertinent drug information regarding the SARS-CoV-2 inactivated vaccines. This presentation will focus on the leading inactivated SARS-CoV-2 vaccine candidates. As of January 27, 2021, there are 12 vaccines in preclinical trials and 9 in clinical trials. There are currently 6 vaccines with registered phase 3 trials. The 4 we will focus on in this presentation are currently in use under emergency authorization in multiple countries outside of the United States. As more information becomes available for the other vaccine candidates, this presentation will be updated. Inactivated vaccines utilize the entire SARS-CoV-2 virus, unlike other COVID-19 vaccine mechanisms, to produce an immune response. The virus is destroyed or killed so that it cannot replicate, but its proteins, including the spike protein and other viral proteins, remain intact. Inactivated vaccines require multiple doses and are formulated with adjuvants to help stimulate the body's immune response to the vaccine. Once administered, the inactivated coronavirus is ingested by an antigen-presenting cell, which then displays some of the coronavirus fragments on its surface. Helper T cells can detect this and recruit other immune cells to respond to the vaccine. B cells can also respond to the inactivated coronavirus and produce antibodies. These antibodies can target viral proteins during an infection with live SARS-CoV-2 and prevent the virus from entering cells. Other examples of inactivated vaccines include hepatitis A and inactivated influenza. First, we will review the preclinical and phase 1 data for Bharat Biotech's Covaxin, or BBV-152. BBV-152 is a whole virion inactivated SARS-CoV-2 vaccine that is formulated with a novel adjuvant. This adjuvant is an imidazopyquinoline class toll-like receptor 7/8 agonist that is absorbed to algel or aluminum hydroxide gel. The TLR 7/8 agonists elicit a strong type 1 interferon response that facilitates the development of Th1 biased immunity rather than a pathogenic Th2 biased immunity. The in vivo animal data illustrated high antigen binding and neutralizing antibody titers at both the 3 microgram and 6 microgram doses in mice, rats, and rabbits. Furthermore, their findings were consistent with a Th1 biased immune response based on elevated IgG2A, IgG1 ratios and increased levels of SARS-CoV-2 specific interferon gamma CD4 T lymphocytes in the Algel IMDG formulation compared to Algel. The phase 1 trial of BBV-152 sought to select 2 formulations for progression to phase 2 trials. In this double-blind, multi-center, randomized controlled phase 1 trial, adults aged 18 to 55 were randomized to one of 3 vaccine groups or the control group, which was adjuvant only. The primary outcome was the number and proportion of participants with solicited local and systemic reactogenicity events at 2 hours and 7 days after vaccination and throughout the full study duration. The secondary outcomes were immunogenicity measured by geometric mean titers and 4-fold seroconversion rate of neutralizing antibodies from baseline to days 14, 28, 42, 104, and 194. 375 participants were enrolled in the Phase 1 trial of BBV-152. 99 were randomized to the 3 microgram Algel IMDG group, 99 to the 6 microgram Algel IMDG group, 93 to the 6 microgram Algel group, and 73 to the control group. Seroconversion, defined as a fourfold increase from baseline, reached greater than 80% across all doses, including the 6 microgram Algel formulation. Participants in the 6 microgram Algel IMDG group reached 91.9% seroconversion rate 28 days after vaccination, which was 2 weeks after the second dose. Seroconversion occurred in 8% of participants in the control group by day 28, suggesting asymptomatic infection. The post-second dose GMTs at day 28, seen in graph B, did not differ significantly between the Algel IMDG groups and the 6 microgram Algel group. The vaccine induced responses that were similar to those detected in serum collected from 41 patients who had recovered from COVID-19. Across all vaccine groups, the most common adverse events included injection site pain, headache, fatigue, fever, nausea, and vomiting. All reported adverse effects were mild, 69%, or moderate, 31%, and were more frequent after the first dose versus the second dose. This is notable since the mRNA vaccines in use now have higher incidences of adverse effects following their second dose. As a result of this data, both Algel IMDG formulations were chosen to move forward to Phase 2 immunogenicity trials. Next, we will review the data from Sinopharm's BBIBP Core V Phase 1 and 2 trials. BBIBP Core V comes from one of 3 strains isolated from 3 patients hospitalized in China. The specific strain was selected due to its ability to replicate and produce high yield in Vivo cells compared to the other 2 strains. The virus was inactivated with B-propionyl and manufactured with aluminum hydroxide adjuvant. This was a dose escalation, randomized, double-blind, placebo-controlled Phase 1 and 2 trial. Phase 1 included adults aged 18 to 59 and 60 to 80 who were randomized to either vaccine or placebo. The vaccine doses were 2 microgram, 4 microgram, or 8 microgram given on day 0 and 28. Phase 2 included only adults aged 18 to 59 who were randomized to either vaccine or placebo. The vaccine groups included 8 micrograms on day 0, 4 micrograms on day 0 and 14, 4 micrograms on day 0 and 21, or 4 micrograms on day 0 and 28. The primary outcome for safety was the occurrence of adverse reactions within 7 days after the first and second vaccinations. The secondary immunogenicity outcomes were measured using an infectious SARS-CoV-2 neutralizing assay and expressed as neutralizing geometric mean titers, and seroconversion was defined as a fourfold increase from baseline. 192 participants were enrolled in Phase 1, with 96 participants in each age group and a median age of 53.7. In both age groups in Phase 1, by day 42 post-second vaccination, there was no significant difference in neutralizing antibodies between the 4 microgram and 8 microgram doses. Additionally, by day 42, the neutralizing antibody titers in the 2 microgram cohort was significantly lower than the 8 microgram cohort. The 4 microgram cohort was not statistically different from the 8 microgram. So, for the phase 2 trial, the 2 microgram dose was not pursued. In phase 2, the 4 microgram cohorts, given as 2 doses either 2, 3, or 4 weeks apart, persisted in neutralizing antibody titers compared to the single 8 microgram cohort. As you can see in the chart on the right side of the slide, the dark purple shaded bar represents the 8 microgram single dose vaccine, and at day 28, the neutralizing antibody GMTs were significantly lower than the other cohorts, with a p-value of less than 0.001. In the phase 1 study, pain at the injection site was the most significant local adverse event reported in the 18 to 59 age group compared to placebo, at 35% in the vaccination groups versus 8% in placebo. This is reassuring as we have seen reports of over 50% of patients experiencing pain at injection site with the mRNA vaccines. Larger phase 3 studies will be needed to see if this trend continues. There were no significant differences in overall adverse events compared to placebo in either age group up to day 28 post-vaccination. There were also no serious adverse events reported within 28 days across all cohorts. In the phase 2 study, more participants experienced any adverse event in the 8 microgram group versus placebo, with 39% versus 11% respectively. The most common injection site reaction was pain in all cohorts when compared to placebo, 16% versus 4%, which again is lower than what we've seen with the mRNA vaccines. Overall, the most common systemic adverse events in the vaccination cohorts were fatigue and fever, which occurred only in 3 and 2% of participants, respectively. Next, we will review the data from Sinopharm's other inactivated COVID-19 vaccine phase 1 and 2 trials, which was developed in collaboration with the Wuhan Institute of Biological Products. The second inactivated vaccine candidate from Sinopharm was derived from a SARS-CoV-2 strain in Wuhan, inactivated by beta-propionolactone and adsorbed to alum. This was a double-blind, randomized, placebo-controlled phase 1 and 2 trial in adults aged 18 to 59. In phase 1, participants were randomized to 3 vaccine groups, low, medium, and high dose, given on days 0, 28, and 56. In phase 2, participants were randomized to one of 2 schedule groups, day 0 and 14, or day 0 and 21. Both phases included a control group which consisted of the alum adjuvant only. The primary outcomes were any local or systemic adverse reaction within 7 days of each injection and neutralizing antibody response, defined as 50% plaque reduction neutralization tests against live SARS-CoV-2, 14 days post-vaccination. The secondary outcomes were any adverse reactions during 28-day follow-up and seroconversion, defined as a 4-fold increase of antibody titers over baseline. 96 participants were included in the phase 1 trial and 224 were in the phase 2 trial. In the phase 1 trial, most participants started to generate antibody responses after the second dose and persisted 14 days after the third dose. In the phase 2 trial, the neutralizing antibody titers were higher in the 3-week vaccine schedule compared to the 2-week vaccine schedule, with geometric mean titers of 247 versus 121, respectively. This demonstrated that longer intervals, such as 21 and 28 days, produced higher antibody responses compared to shorter intervals, such as 14 days. Additionally, seroconversion was seen in more than 97% of all participants in phases 1 and 2. The most common adverse reaction was injection site pain and fever. The total adverse reactions were similar across the 3 vaccine groups in phase 1. More participants in the medium dose 0 and 21-day group experienced adverse reactions compared to the 0 and 14-day group, with 14% having injection site pain compared to 2.4%. However, in general, the incidence rate of adverse reactions among all participants was only 15%, indicating that it was well tolerated and lower compared to other candidate vaccines. Next, we will review the phase 1 and 2 data for Sinovac's CoronaVac. CoronaVac is an inactivated vaccine candidate created from African green monkey kidney cells, or Vera cells, that have been inoculated with SARS-CoV-2 and inactivated with beta-propionolactone and absorbed onto aluminum hydroxide. This was a single-center, randomized, double-blind, placebo-controlled phase 1 and 2 trial in adults aged 18 to 59. Participants were randomized to either 3 microgram or 6 microgram doses given either 2 or 4 weeks apart. Phase 1 was a dose escalation trial where participants were recruited and allocated to one of the 2 vaccine schedules. Within each cohort, the participants were assigned to low dose 3 microgram or placebo and followed for 7 days after the first dose for safety. Then, the next group of participants were assigned to the high dose or placebo. Phase 2 begun once all participants in Phase 1 had completed a 7-day safety observation period following their first dose. Similarly, Phase 2 participants were recruited and allocated to one of the 2 vaccine schedule cohorts, and then randomly assigned to low dose, high dose, or placebo. The primary safety endpoint was adverse reactions within 28 days after injection. The primary immunogenic outcomes were seroconversion of neutralizing antibodies to SARS-CoV-2 at day 14 after the last dose in the day 0 and 14 vaccination cohort, or day 28 after the last dose in the day 0 and 28 vaccination cohort. 144 participants were enrolled in the Phase 1 trial, and 600 participants were enrolled in the Phase 2 trial. The investigators noted the significant difference in immune responses between Phase 1 and Phase 2. They discovered that due to a change in their manufacturing process, the vaccine formulation used in Phase 2 contained a higher volume of intact spike protein. In Phase 2, by 28 days after the second vaccine dose, the neutralizing antibodies to SARS-CoV-2 were not statistically different between the 3 microgram and 6 microgram dose, but the 4-week vaccine schedule appeared to elicit a higher response. There were no significant differences in adverse events among the different doses and schedules. Overall, the incidence of adverse events was 20% and 25% in Phase 1 and 2, respectively. The most common adverse event was injection site pain. with 13% in Phase 1 and 15% in Phase 2. More participants reported this in the day 0 and 14 schedules. There was one cause of hypersensitivity with urticaria in the Phase 2 study that occurred 48 hours after the first 6 microgram dose. It was determined that this possibly was related to the vaccine, but no similar reaction was seen after the second dose. There were no serious adverse events reported within 28 days of vaccination. While there are no inactivated COVID-19 vaccines currently available in the United States, there are some promising candidates that are already in use in other countries. These vaccines have the potential for decreased local and systemic reactogenicity compared to mRNA vaccines, and their platform has historically been suitable for use in immune-compromised populations. A theoretical concern exists for antibody-dependent development of infection if a person subsequently becomes infected with the virus, which highlights the importance of determining Th1 response during these Phase 1 and 2 trials. Lastly, as with many current vaccine candidates, inactivated vaccines require at least 2 doses for efficacy with the potential for periodic booster doses. Information regarding COVID-19 vaccines and other frequently asked questions can be found in the resources listed here. Thank you so much for watching. This has been a review of the pertinent drug information for SARS-CoV-2 inactivated vaccines. This presentation will continue to be updated as more information becomes available. If you have any questions regarding the material in this presentation, my contact information is listed here.